Okay, Peggy, you're on.
Good morning, everyone. My name is Peggy Munson, and I'm an AARP volunteer for New Knowledge Adventures here in Boise. And New Knowledge Adventures is a volunteer-driven program sponsoring all types of classes for education and just plain fun. These classes are offered in person, online, and through the VCC. Today, we're going to ask you to go into the chat room and let us know where you are from so we can know how many people across the nation is attending this class. So please go to the chat room and tell us where you are from. And it is my great pleasure today that NKA is welcoming Dr. Greg Hampikin, who is a professor in the Department of Biological Sciences with a joint appointment in the Department of Criminal Justice at Boise State University, as well as the University of Idaho. Being an established professor with over 20 years of teaching for various departments, Dr. Hampikin teaches a variety of insightful classes, helping bridge criminal justice and science together. In addition to his substantial work in higher education, Dr. Hampikin has been a director and leader in various projects and foundations. And most notably, he is the founder and co-director of the Justice Forensics Project at Boise State University. Through his work and efforts, his groundbreaking research in forensic DNA technology has not only advanced the field, but has contributed to the exoneration of more than 36 wrongfully convicted individuals. So go ahead today and let's welcome Dr. Hampikin.
Thank you very much. Can you guys hear me okay here? I see we have a great crowd. Thanks for nodding, people. They've muted everybody. If you have questions, we're going to try to take those during the talk because I really like having a live classroom. I don't like taping these type of things. So I appreciate your critical thoughts. My students, when they go to a talk like this, they get extra credit if they ask a question of the speaker. Twice as much credit if the speaker cries. And that's because what I'm trying to encourage in my last years is critical thinking and using reasoning, applying the frontal lobes that kind of distinguish us from everything else for light years, probably, from where we are. And that's what we're going to do. So one kind of disclaimer is I'm going to be talking a lot about mistakes in forensic science because that's my kind of focus. But I don't want to give the impression that it's garbage or that we shouldn't trust labs or shouldn't trust police or the courts or something. I do trust them. I work within them. I'm going to be testifying tomorrow. I'm watching another trial and a case that I helped work on. That's a 43-year-old case where a child was raped and murdered. So I am heavily invested in the system, and I support the system. I criticize human beings, myself included, the way that we reason. And that's what I'm going to try to show you. So maybe you can avoid some scams, or you can look at some of these crime shows with a larger depth of critical thinking. So I'm going to go to my PowerPoint. And PowerPoint's kind of a necessity for me in going through this, but I'm happy to be interrupted, as I said. Whoops, let me make sure I've got my sharing on. I'm going to share this PowerPoint. Let's see if it works. All right. Can I have a thumbs up if all you see is that first big slide? Awesome. Thank you, Sheena. You are the one who pops up. So, okay. We're called the Idaho Forensic Justice Project. And I know in the advertisement, they mentioned the Idaho Innocence Project. After 20 years or so of pursuing that here in Idaho, I decided that we are not going to do a full legal representation anymore. We're going to do just focus on the science. And that's why we're calling ourselves the Forensic Justice Project. So the Idaho Innocence Project has ended just a couple weeks ago. And we are forming an alliance with a group in California. And I hope to have all of our cases transferred to those fine lawyers. There'll be a lawyer here in Idaho working on working on Idaho cases. But we're going to focus on science. And so my talk today nicely focuses on science. I'll talk about some of the folks who were freed through the work of the Idaho Innocence Project at Boise State that I founded and co-directed for many years. Here's some of the folks who've done work on these cases or experiments in my lab. Some of them are active, some have moved on, but this is some of the recent staff. In case any of you know any of them, you might give them a hard time saying that I showed their slide. And here's a gentleman who I met for the first time in New Orleans a few months back, Terry Tolley. And I had lived in Georgia, so I worked on a lot of Georgia innocence cases. I was on the founding board of the Georgia Innocence Project. I did their first DNA case. But Terry's case came to me after I'd moved to Idaho, and I was asked to review some DNA results in a rape case. And the great news was the results were clear. They'd finally gotten DNA done, and Mr. Tolley was excluded. And I thought, great, I sent my report to the folks in Georgia, and I said, so is he going home soon? And they said, not quite, there's some problems. It's like, what do you mean problems? What could possibly be a problem? And the problems were that there were 4 cases, one of which he had pled guilty to or pled in some way. And we had only undone one with DNA. Now, this is an example of some prosecutors getting together with good lawyers from the Georgia Innocence Project. And after that first case was undone, realizing that all of these cases are threaded together by that initial error. It's kind of, when you get a bad reputation, and being known as a rapist or murderer is a bad reputation, you become one of the usual suspects. And there is a lot of pressure to solve cases. And so you take the most efficient route, and sometimes that means the easiest route. And Mr. Tolley was railroaded in all 4 and finally is freed after 39 years of incarceration and some great work by the Georgia Innocence Project. Now, when you've been in for that long, it has a very obvious impact. And in talking with Mr. Tolley and talking about work, he had to get a job in his 60s for the first time, or first time since he was incarcerated. And you can imagine his discomfort with technology. When he went in, cars didn't even have sound effects, let alone voices that talk to you. So much had changed, and it's a pretty typical and horrible experience for these people. And while it's great to see someone walk out of prison, as soon as you remember that they should have never walked in, it takes some of that exuberance out of it. And being in this. My experience is mostly working on cases with frustrating slowness, and the exonerations are, tips of the iceberg. They're great, but they're really not the typical day. Okay, another man's case I worked on. This is Stephen Pina. He is free in Massachusetts. I think he still has an ankle bracelet though, because they're deciding whether or not to try him again for a murder conviction that was undone by some good folks at the New England Innocence Project, and I had worked on the DNA aspects of that case for them. So I also met him in New Orleans. We have a conference once a year at the Innocence Network. Gilbert Poole, actually, my partner Janet Lane had worked on his case, and I worked with her, but really she was the one who was doing the work. And Gilbert was in for 32 years. false conviction of murder, freed in 2021. He sells T-shirts now, so if you look up Gilbert Poole, Poole, you can order some T-shirts. My philosophy going into all these cases, whether I am hired as a private consultant, and that generally happens for the defense when someone is accused by the state of something, or when I'm getting a letter from somebody who's in prison who says that they're innocent, is this statue from outside of the Connecticut State Police Forensic Lab. Henry Lee had this put up, and Henry's one of the giants in our field, really pioneered a lot of the DNA techniques. But it says that if the law has made you a witness, remain a person of science. You have no victim to avenge, no guilty or innocent person to convict or save. You must bear the testimony within the limits of science. Let the evidence speak for itself. And as clear as this sounds, as a human being, I think it's impossible. I don't know anybody who's able to dispassionately work on something for 10 years, present the case before a presiding judge who's going to decide the fate of someone you either think is a child murderer or is innocent and is a victim of the state. It's impossible to remove bias, though I really try to remember this each time. And I'm fortunate that I have a partner in life who will argue cases with me back and forth, like a prosecutor or a defense attorney, on the opposition. And that critical thinking, and all thinking in terms of science is critical. It unfortunately doesn't have a lot of platitudes. That critical thinking is really important. I'm going to try to work through some of it with you guys. So let's define science. And I worked on Amanda Knox's case a little bit in Italy. She had a great team on the ground, but I worked with that team for several years. And so I developed a slide that features an Italian scientist. These are quotes from Galileo. Galileo really helped define the scientific method as I see it, the experimental scientific method. So let's take a few of his premises, and you can apply this to your own life. So by denying scientific principles, one may maintain any paradox. So what is a paradox? Paradox is two statements in opposition that are both claimed to be true. So do we do this? Yes, we do it all the time, right? You're allowed to do it in religion. You can do it in love for a child who's, constantly doing the same error over and over again. You can maintain paradoxes in many parts of your life, and they're useful. Hope is a kind of a paradox. But in science, you can't do that. You've got to resolve your paradoxes, or at least admit them. In questions of science, authority, the number one fallacy, the authority of 1,000 experts he's talking about is not worth your humble reasoning. Your humble reasoning. And I've seen people defend themselves in court and convince a judge that everyone got it wrong. The police got it wrong. The prosecutor got it wrong. And they do it by reasoning. Count what is countable, measure what is measurable, what is not measurable, make measurable. And I study novel sequences in evolution that come up, and I also ask people about novelty in their own minds. I've walked around Boise asking artists and mathematicians and anyone else, what's the smallest unit of creativity? Because I want to measure it. I want to know, what's the difference between something that's derivative and creative. Measure it, and you can put it into a formula. You can relate it to other things, but you've got to be able to measure it. And a lot of times in these crime shows, what you're going to see is, I had a hunch, right? You'll hear that all the time. This detective had a hunch. The evidence was pointing another way, but the detective had a hunch. Yeah, I don't believe that stuff. I don't believe it in the slightest. Hunches are the substance of witch hunts, of prejudice, of hope, but not of science and not of reasoned convictions. Science proceeds more by what it has learned to ignore than by what it takes into account. I know that they sell shampoo with DNA in it. I know that DNA is not going to help protein in your hair, so I don't buy it. Science has taught me to ignore certain labels, health claims, because I'm trained in the methodology and can spot some of the obvious charlatans. That's all from Galileo. So if you keep these in mind, you're going to watch these shows and trials a little differently. I brought up recently, and people on my staff often get upset with me for bringing up the O.J. Simpson case. But the day I brought it up, I was giving a talk like this, and then O.J. died that day. And so I went back and watched a lot of the videos. And I can ask you, folks, how many people think O.J. did it? And I can ask you how many think that the jury in his criminal trial was racist or wrong, reverse racism, whatever you want to say. And a lot of people will weigh in on that question. I just had a colleague do it at dinner a week ago. She said, well, that jury, they didn't consider the evidence. And I said, excuse me, I spent a lot of time in a courtroom. Courts are very boring places. It's very hard to stay awake. That jury in OJ's case was sequestered longer than any jury in the United States, 11 months. People I know who say they watched the trial, I say I watched the trial. Maybe I watched, 20 hours, 40 hours. I didn't watch 11 months. And those people watched it, and they came to a reasoned conclusion. It only took them 4 hours. When a jury stays for 11 months in a case and takes 4 hours to make a conclusion, that jury is very sure of what they're going to say. And I respect that. I want you to use your reasoning like people in a jury have to do, and realize that the press are storytellers, and the better the story, the more sensational, the more they can get people riled up, the more they sell. We are stimulated by that type of thing. And I remember when the OJ verdict came out, and my Black friends and white friends had totally different reactions. And that told me, oh, this is not about reasoning, right? These are all smart people. Something else is going on. Okay, so how do we measure the success of something as complex as the criminal justice system? Well, it's an election period, and candidates will tell you they're tough on crime. But what does tough on crime mean? It means we have to have a system that somehow penalizes tough on the criminals. So how do we measure that system? Did we get the right guy? And I'll use guy because the prisons are quite gender imbalanced. There's many more men in prison, especially for violent crimes. So did they get the right guy? Is that a good measure? Ask yourself if that's a good measure of the criminal justice system. They got the right guy. And so this is what I developed to try to explain to people what we really have to do in science. If we measure the justice system by the number of times they really get the right guy and convict him, then the Salem witch trials were an astounding success. They got more than 100% of the witches. More than 100% of the true witches were killed. So success is not the way we measure things in science. You don't know if you have success until you determine your error. In some manner, you have to look at error. You have to decide what it is you're measuring and where your measurement is wrong. When does your speedometer fail? You can't use your car speedometer to track the speed of an inchworm. It just doesn't work. It doesn't work at that scale. All right, so what I did, finding out that very, very few people were studying this back in the early 2000s, is I got together with some folks at the Innocence Project in New York. They did a lot of the heavy lifting on this paper. It was my idea, and so I'm the first author. I don't know if that's fair, but they really did the great work. Going through all of the exonerations up until that date in 2010, 194, and saying how many times was it a cigarette butt? How many times was it something we call mitochondrial DNA? How many times was it just male DNA, Y DNA? when people were freed from prison. The reason I started doing this was because I'd go to court with a group of people who want to test a cigarette butt from a crime scene, or they want to test fingernail evidence. And judges would say, well, has anyone ever been freed from a fingernail? And I didn't know. I didn't have numbers. Well, here you can see fingernail evidence was 5% of the exonerations. Cigarettes were 3% of the exonerations. What am I doing? What were we doing? Count what is countable. What is not measurable, make it countable. And we hadn't had a record of what exonerations were about. So this was part of an early part of this movement of trying to study the science. And research is the biography of science. It's what's already been done. So I'm going to introduce one of the problems. And here's where you got to really put on your thinking caps, because this took me 30 years to figure out, but I think you're going to get it quicker. In a courtroom, the state is generally putting on evidence, and the defense is countering that evidence and then putting on some of their own evidence. But the state's burden is to present evidence in order to convict. And what if there isn't evidence that shows that someone was at a scene? So DNA is often used to say, we found Greg's DNA on the knife, or we found it in the room where the body was, or we found it on the body. And that DNA provides evidence that I was there, or that somehow my DNA got there. When my DNA isn't found and I'm accused of a crime, this statement often comes up. Well, the absence of evidence is not the evidence of absence. Just because Greg's DNA is not in the room with the body, that doesn't prove Greg was not in that room. The absence of evidence, you can't prove a negative, is the way the philosophers might say it. Sounds right. I've been asked this on the stand. Dr. Hampikian, you said there's no evidence, of John Smith's DNA anywhere on anything there, and there's evidence of someone else. But that doesn't really mean he wasn't there, right? Because the absence of evidence is not the evidence of absence. And I would say yes. Well, what do you think? Is there anybody who wants to raise their hand and say there's something wrong with that? I'll try to look. What could be wrong with that? It sounds perfectly sane. It's perfect reasoning. Just because there's not evidence doesn't mean the person was absent. So here's how it's used in court. This is a sexual assault case. There are 4 vaginal swabs. So they take a Q-tip and swab the complainant, the person who says that they were a victim of sexual assault. And in this case, there was insufficient male for further DNA processing. Insufficient male DNA. That's what this says. So what does that mean? Well, this lawyer, who was really smart, he's an eclectic Princeton graduate who plays in the orchestra and was raised by hippies, a hippie single mother, as he proudly says. Anyway, he asked me, parse out this sentence for me and tell me what it means. Extracted and quantified insufficient male. That means they got some male DNA, right? They're going to use that against my client. They got it, right? And so I went back to the records, and I've got the actual readings from the machine that measures the quantity of DNA, and it said 0. It said 0 male DNA. Now that's truly, it's insufficient, but it's actually a test. In other words, if I'm accused of a crime, the way that I put this when I talk to the analysts from all over the country, when I get to give one of the big talks for our scientific meetings, recently I've been trying to give them this analogy. Think of your favorite young male relative, somebody in college. They're going to go for their year abroad, like Amanda Knox did. They're going to study overseas. And they go to a country you've never been to. You don't know anything about it, really. And they write to you and say, I took a babysitting job. And you're like, great, you're getting a little money. And then they write and they say, I've been accused of molesting the child. And I swear I wouldn't do anything like that. And then they call and they say they've taken DNA from me, and that they're going to swab the child and the diapers, and where the assaults supposedly happened on a change table or something. And you wait with bated breath, and they come back, and there's none of your nephew's DNA or your relative's DNA, any of those places. Well, what do you say? Hooray. My God, there's evidence. But what the prosecutor might say is, well, they'll ask the DNA expert after they say, we didn't find his DNA anywhere. Well, the absence of evidence is not the evidence of absence, is it? So what is wrong with this phrase that is used all the time in the court? What is wrong with it is that the, the absence of evidence is in fact evidence. If you take a pregnancy test and the test is a good test and the control works, and it says you're not pregnant, that's the absence of a fetus, the absence of human chorionic gonadotropic hormone. That is absence, and it's evidence. Now, it's not 100% certain, but nothing in life is 100% certain. A positive test is not 100% certain. Somebody could plant it. The test could go wrong. There could be contamination. So this is part of the innocence penalty that is throughout the system. The system evolved to get the bad guy. It accidentally also eliminates a lot of innocent people, but the system itself is full of language that is biased toward getting the bad guy. And this is one of them. The innocent person has nothing but the absence of evidence, right? I wasn't there. Test everything. Look at the videotape. Look for my fingerprints. Look for DNA. Get any audio you can. Talk to the neighbors. I wasn't there. Absence, 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 absence. I mean, I wasn't there, right? No, because the absence of evidence is not the evidence of absence. And that's what they'll say. And experts like me nodded for years to that. And I'm trying, this is one of my things for this year. I'm trying to break the system up. We'll see. Okay.
We had one person, Dr. Hampikian,
Yeah.
that had the nerve to say, "Thank you, Sherry," because if it's true, the absence of evidence is, then you could accuse anybody of being at any crime.
Exactly.
That is exactly the opposite of what we're trying to do. But you could
not only can you accuse them, you can actually use it against them that there's no evidence. You can actually discount all of that study that was done by the state. And the state, what this totally masks in I think, a very cruel way, is that the state had a hypothesis. They thought your nephew was on that child. They were testing a hypothesis. They took DNA from your nephew, DNA from the child. The hypothesis was, well, we're only comparing one person, your nephew. The hypothesis was the nephew contributed DNA to the child. When they get a negative result, that's a negative result, but it is a solid result. Just because it isn't what we wanted, it's still a result, negative or positive. Okay, so whoops, let me try to go on from here. Negative results are the evidence of absence. Absence of an embryo. If the doctor says you don't need a mastectomy after a biopsy, that's the absence of cancer, the absence of viral DNA that allows you to stay home for a week with COVID. All of these are tests. The crime lab is testing a hypothesis about foreign DNA. The hypothesis is your nephew left the DNA on the swab. If the controls work, no foreign DNA is found. That is a valid refutation of the hypothesis. It is an exclusion, but no DNA expert uses that word exclusion unless they're comparing two profiles. If they're comparing your nephew to a valid test that showed no foreign DNA on the child, they won't say he's excluded, some labs. They'll just say, no male DNA was detected. Like that picture I showed you, insufficient male DNA. They don't say he's excluded. We looked for male DNA and he's not there. Somebody has a question, I see.
Yes. Bill, you have a question that you wanted to ask?
Yeah, thank you. I had to request permission to be unmuted first. So, Professor, I'm obviously I don't work in this field, so I'm not used to these phrases.
Yeah.
So I'm going to remove myself from the use of the phrases and just use my everyday common understanding of things. Uh, I don't know about Italy, but in the United States, the burden of the state is to prove guilt, and you are innocent until proven guilty. So if the state goes in and finds DNA, they've got some evidence that they can use to move towards a proof of guilt. If they go in and they find no evidence, no DNA, no anything, we all pack up our bags and go home. What am I missing here?
Well, yeah, I'll show you what happens in actual cases. Let's see. Oh, sorry, I just have to respond to a request for testimony. That would seem like that's the way it's supposed to happen. And in fact, when I was interviewed about, Amanda Knox's case, I said that too. When they looked for her DNA and found absolutely none of it in the first round of testing, and they found someone else, and that guy's even in the victim's on her body and her vagina, and his handprints and blood on her wall. Once they found that, they probably should have let Amanda and Raphael go. But that's not the way things happen. When we are invested in something, it is a commitment. And I tell my students, for years I had thought maybe I want to go into theology. Now I realize no religious body would have me probably. But and then I thought about going into science. And my friends who went into theology are rightly judged by their willingness to die for their beliefs. And they really would. They've given their lives to these beliefs. And the measure of a scientist is how quickly you'll let your beliefs die. You do a test. I've developed cancer drugs, right? I have not cured cancer in mice. I have to believe my results over and over again. 20 years working on these drugs. But the mark of a good scientist is to go into lab meeting. You've paid all these people, and you've had them work, and you've put all this time into something, and you have to look at their data and say, well, there's nothing here. And that's what science should be about. That's not what I see in the legal system. So here's how the typical exchange goes. This is from an actual case I'm reviewing. Prosecutor is asking the state's DNA analyst, okay, they didn't find any DNA from the suspect. Okay, when we don't find somebody's DNA, does that mean they weren't at a particular scene? And this sounds kind of rehearsed to me, but anyway, answer, it does not mean they were not there, says the DNA analyst, just because you don't find the DNA. Okay, so the absence of evidence is not the evidence of absence, so to speak. Correct. Thank you. That's all the questions I have. And the defense lawyer did not really do well on this crossing. But here you see it. You get in the court, and I've been in court with people who are innocent or eventually found innocent, and they're shocked. They go in and they're like, they have someone else's DNA. Why are they prosecuting me? And they really think the jury's going to understand this, like you thought. But it's not the way it goes. So the FBI now puts this footnote at the bottom, in 2019, puts this footnote about DNA, that number 2 footnote. Insufficient DNA, quality or quantity, can affect the ability to generate a DNA typing result and is not an absolute determination that an individual did not come into contact with an item of evidence. Sound good? Yeah, now they're saying it's not they're not saying it's not evidence. They're saying it's not an absolute determination. Do I have a problem with this? Yes, I do. Of course I do. That's why I'm showing it. I want you to think about what the problem might be. This is an entirely true statement. Is there anything you would want added? I would suggest something like contamination in planting can affect the ability to generate a DNA typing result. Therefore, a positive result is not an absolute determination that an individual came into contact with an item. And I wouldn't say just those two things. I would say storing items together, people who live in the same house, now even dust, our tests are so sensitive, can imply that someone touched something when they had nothing to do. They could have been dead and their DNA is transferred, right? Abraham Lincoln's DNA is on a lot of things that he never touched. It's people handled that chair that they've preserved full of his blood. They pushed that DNA other places. They've been handling it, for over 150 years. Okay, so positive test. Is it an absolute determination that someone came into evidence? So why don't you add that too? It's still this bias of get the guy who did it. And that, when you call the police, you don't say, hey, I've been robbed and attacked. Make sure you eliminate all the innocent people. They're like, go find who did this. And so I understand why there's bias. Does this actually happen? Am I just talking about philosophy? This is Christopher Tapp, who was a very good friend of mine. And we worked on his case a dozen years or so. And he got cleared when we took a novel strategy, even though there was DNA at trial that did not include Chris. So there was a woman who was raped and murdered. Somebody left semen on her body. The semen DNA did not match Christopher Tapp. There was a complete profile, but it didn't match Christopher Tapp. And they convicted Chris. They got a false confession out of him, promising he would do no time in prison because they thought his friend did it. So he just had to say his friend did it. And that's what he said. Oh, I was there. I watched it. I did it. I stabbed her once at the end. That's because they had to have him, part of the crime scene. And, and I held the teddy bear on her and but this guy did it. And so they went and tested his friend, which is what they wanted, probable cause test friend, and he didn't match the DNA either. So they rescinded their deal with Chris and tried him and used parts of that confession as the court allowed. And what did his lawyer, who I thought did a pretty good job from the transcripts, his lawyer said to the analyst, well, you have somebody else's DNA, right? Yeah. It's semen, right? Yeah. It's on a dead woman. Yeah. Do you have any of Chris's DNA? No. And they thought the jury would acquit him, and the jury convicted him. And why? Because the prosecutor said the absence of evidence is not the evidence of absence. It happens. This is real, and it's witchcraft. It is not science. It is not reasoning. But people like me, I'll just say, smart people like me, got fooled into thinking this was true for years because the training in the system is about get the right guy. Okay. So we got involved with forensic genealogy. There's a lot in the news about that now. We have the Kohlberger case here in Idaho. We have what I think is even more important a case. Dalrymple is on trial right now in Canyon County, Idaho. It'll be the first case anywhere that uses the kind of DNA that genealogists use, single nucleotide polymorphisms, they're called, or SNPs. It'll be the first time that's ever used in a criminal case. So all these cases where you have leads generated by genealogy, we find DNA, as in the case with Chris Tapp. There's DNA, we don't know whose semen it is. There's no matches in the database, in the criminal database. So what did we do? Well, I came up with a method that other people had talked about, but we were the first ones to really use it. And that was to take DNA from a crime where someone had been convicted and look for the male component in that semen, the male DNA. So we worked with the Idaho Falls Police Department. They thought Chris was guilty, but maybe another guy helped him. And so we found the last name of the semen of the father of the person who left semen. We found a genealogical match, and we did it starting in 2012, before this became kind of a national strategy. Other people were trying things. We tried this particular way, and it worked. We got a name. And that work was finished then by C.C. Moore, and I think she's got a couple TV shows about it. But it led to an actual perpetrator in Chris Tapp's case. So not only did we got him freed based on the fact that we tested all the DNA in that room, the teddy bear that Chris said he had handled, the shirt of the victim, we tested everything, and it was all one guy, not Chris Tapp. We found the last name lineage of that guy, and they developed a full genealogy from that, and eventually arrested a man. So the name we found was Ussery. That was the father of this guy. His mom did not give him the biological father's name. So Cece and her team of volunteers had to do a lot of work to find a guy named Brian Drips, who was the son of a man named Ussery, the name we came up with the Idaho Falls Police Department. Okay. But remember, Chris was convicted on the absence of evidence, not being able to establish his innocence. So that's why I put that, be careful about that whole absence of evidence thing. This is my first case in Georgia when we founded the Georgia Innocence Project, or they found it and I got on the board. And the client would, draw us pictures and write us things. And I like to show this because a lot of the people here are at least as old as I am, and so we know some stuff about life. But the thing to keep in mind is you can't tell who's lying to you. And all of this nonsense about I'm a good judge of character, they've done controlled studies with detectives. They're no better than flipping a coin at telling whether someone is lying or telling the truth. I have a book outside of my office, an advertisement for a book. It's called Deception, an Essential Adolescent Skill, I think is what it is. And you think about that. You have to be able to adjust yourself to your circumstance, right? You're going to behave differently in court, hopefully, than you are, with your buddies, your friends. And every year we run a national test of this system. We inoculate the population with this concept that you do not know who's telling you the truth. And it's where we take all the children, and the National Aeronautics Association, NOAA, gets involved, and the newscasters, and there are people staged outside of shops all over the place. And we tell the children that a fat man in a red suit's going to come down the chimney. We also tell them that a ferry is going to deliver quarters for tea. All of this is very important. I don't think deceiving children is a bad idea, because how did you figure out there was no tooth fairy? How did you figure out there was no Santa Claus? How did you figure out everybody, your closest loved ones, were lying to you? It was a conspiracy of the grandest scale. The entire world is in on this, right? And that teaches reasoning, that you need not to believe people. We should have more of those things, right? It turned out that this guy was either lying to me or somebody planted the evidence, because the DNA agreed with his guilt. And it was on the front page of the papers of the first case the Georgia Innocence Project did. And it was kind of hard for the people who ran the project to get that bad publicity. But I thought it was great. We got an answer. The science worked. Okay.
Dr.
Yeah.
Greg, we have one question here.
Please.
Darlene, you have a question that you wanted to ask before we move to problem 2?
Yes. When did this argument, absence of evidence, become an established argument? Was there a case that made that be an acceptable argument?
Thank you for that, because if you're going to go and Google this and get the answer, please email me. I'm really easy to find. My cell number is even on the web. But I tried to find the first instance, and I didn't spend that much time, and I couldn't. And there are references to ancient philosophy about this, that you can't prove a negative. But I'm not sure how it became so entrenched. Certainly when I started in forensics in the 1980s, it was well established by then.
Wow.
I'm not sure how it's still used today. All over the country it's used, and the experts nod their heads.
Yeah,
mean-
because it usually takes a case or something to make something an argument. So I'm wondering.
It worked for somebody, and it, you know, I mean, imagine you're the prosecutor and you're going into trial and you have an absence of evidence. You're like, God, how do I make this work? Ah.
Thank you.
You know, oh, you can't prove a negative. I'll spin that one. And yeah, I don't, you know, I think maybe the first person who used it realized it was disingenuous. But as soon as these axioms become established and people start using them in the system, they're laws. Everybody gets taught them. Nobody's going to go against it. I tried to reason this out. Like I said, I give these talks to like 300 DNA experts at a time, and I asked them, what's wrong with this? And they really couldn't get it at first. I think I convinced them with the story of the nephew. I hope so. All right, so here we're going to talk about, this is a new term. I'm just going to use the official term, activity level propositions. What does that mean? DNA can tell us who left some biological material, who was the original source. It gives us a lot of great information about the who. It can't tell us how. And yet there are experts like myself, but different, who would get on a stand and say, there's DNA on the trigger of that gun. I can tell it wasn't because it was stored with the clothing of this person or in their car. You know, it was no accidental thing. There's enough DNA that I can tell they handled it. And to those people, I say, really, you can tell that someone touched something rather than there was some other sort of transfer? Yes. I said, well, can I have your toothbrush? Because the idea is I can take your toothbrush. There's enough DNA on your toothbrush to look like any crime I can think of. There's plenty of cells on that toothbrush. And so you can't tell by the amount of DNA how DNA got somewhere. And really, DNA doesn't leave any story about how it got somewhere. You know, there may be some very fine things that one can do in a research lab, but really the crime labs can't do any of this stuff. And so what about this term? Has anyone heard of the term touch DNA? Raise your hand if you've heard of touch DNA. So this became popular in the Ramsey case, the little girl who was in beauty pageants, and we've all seen the photos if you were around in the 90s and 2000s. And they reopened her case because they had her underwear. And in the old days, we used to look for a stain. In fact, there was something called the crust test, where you would take underwear with your bare fingers and rub it to see if there's enough crust on there from semen, from some sort of drainage from the victim, to get DNA to do a profile. You needed a lot. And believe it or not, I've had to go back to some of those cases where somebody's claiming innocence and find out who the analyst was who touched that crotch so we can eliminate them, you know, with these tests. They weren't wearing gloves. But anyway, what happened about 2005, 2010 was people started using this term touch DNA. And what does that mean? It means there's no crust, there's no stain, but I think somebody pulled these down, or somebody ripped this off, or somebody touched this piece of property or person. And I'm going to swab and look for a few cells, try to get DNA. And that started being called touch DNA. Okay. Anybody have a problem with the term touch DNA?
So if you took my toothbrush and touched it to something, does that now equal touch DNA?
Yes, and also, you know, these terms are ancient to all of us. Touch. We were all taught that a stranger could touch us inappropriately. Did he touch you? You know, did you, you know, or if you hit some, you know, your sister, did you touch your sister? I didn't touch them, right? Touch always meant I touched somebody, I poked them, I grabbed them, I whatever. It means your hand. We don't think of touch as my, you know, my little sister picked up my sneaker and she touched the inside and she has my DNA now on her hand. So that's touch DNA from me. That's really a bad use of that word, but that's what that word was being used. So anywhere that people found a little bit of DNA was being called touch DNA. And it was presented to the juries as touch DNA. And we don't know how it got there. It could have got there from your toothbrush, your hairbrush, whatever. And the other thing, just to let you know, I mean, why should there be a lot of DNA in your hands? You wash your hands all day. You don't wash your forehead. You know, you don't wash your chest all day. But the thing is, if I scratch myself or I wipe my hair back or pick at my teeth or whatever, and all the things we can do with our hands, we're getting the sloughed off cells from around our body. And that's probably what we're transferring. So think of that. That is not direct touch. Anyway, that's coming from somewhere else, being carried now by the fingers. It's indirect. And so even though we call it direct touch, it's really not true. It's coming from somewhere else most times. It seems, anyway, the evidence seems to be that. So I've been working with the labs on abandoning that term, and I think I think we're getting good progress on there. But here's how this is used in a court. These activity level propositions. So I get a disclosure if I'm on a federal case and there's an FBI expert going to testify, and I've been hired by the defense to review the case or to work on the case at trial. And this is a disclosure I got, in 2022. The FBI expert will further testify. I've just crossed their name now because I don't even know that the expert agreed to this. This is given to me by the federal lawyer. And sometimes the lawyers just write down what they think the expert's going to say. Now they're supposed to get people to sign off, I've been told. Anyway, the expert will further testify that the levels of DNA, the amount of DNA, the number of cells of DNA found on the firearm are consistent with the defendant having actually handled or touched with his bare hand or skin. No. So, okay, what's wrong with this? Well, first of all, there's no way to tell, as I tried to tell you. And I think any expert would say the toothbrush analogy works. But also, there's a word in here that we've been told for years not to use by the Department of Justice, consistent with. What does that mean? That means anytime male DNA is found on a rape victim, it's consistent with me having attacked that person. Everywhere in the world that they just, the first test they do is how much DNA, is it male or female? As soon as it's male DNA, it's consistent with me. That word is meaningless because it's not associated with a number. The only number it's associated with is not 0. There's not 0 chance that Abraham Lincoln left some blood on this gun. It's, not 0. I told you there's a bloody chair. It could have been transferred somehow. So this is the kind of garbage, frankly, that I have to deal with that is entrenched in the system. This is an FBI expert in a federal firearms case, and I get official notice that this person has the ability in their mind through quantitation, through quantity of DNA, to say what happened. And I'm like, that's remarkable. All right, so where is DNA now? I'll give you a little basic rundown. Many of you have seen a picture of a cell. There's the nucleus in the middle. There are also these mitochondria. They're actually much smaller than this, but this is just blown up. And the mitochondria, help us use oxygen to burn carbohydrates and other energy sources. They came probably from bacteria that infected our species long ago, and they have little circles of their own DNA in it. So sometimes, because there can be 1,000 of those mitochondria in a cell, sometimes that's all we have left with skeletal remains or bodies that have been burned, or very degraded DNA that's been left out in the sun or something. But most of the DNA that we like to get is this chromosomal DNA. You get half from your mom and half from your dad. 23 chromosomes in a sperm, 23 chromosomes in an egg. Now you have 23 pairs of chromosomes. And that's what those blue strings are. It's kind of a very cartoonish representation. So how do we get the DNA? Well, just a swab, which is basically a Q-tip. They're made of cotton or polyester. And these are cells stained with methylene blue, probably from someone's cheek, if you've ever done a high school cheek staining. And you can see that these cells, they're called epithelial cells. They're the same that's in the cheek, the vagina, the penis, the anus. It's pretty much what we see, except for, blood cells or bone or hair. And they break very easily. You add a little bit of detergent and a little meat tenderizer, a proteinase, something that dissolves protein like a meat tenderizer. And you can break open those cells and get the DNA out. Okay, when I first saw this many years ago, it confused me, but let me try with you. This is a cell. This is that cell I was showing you all the way up at the left. Has a lot of mitochondria in the cytoplasm, not the nucleus. And it has the 23 pairs of mom and dad's chromosomes in the nucleus. And if I use a microscope, that can blow up 500 times or so, I can see the chromosomes. So this is a blow up. This green arrow means magnification. And there's a chromosome there. And then if I could go another 100 times or so, I would see the DNA strand that's wrapped up in this chromosome covered with proteins. And then if I could blow that up further with a really good electron microscope, I might see members in the helix, actual bases, A, G, C, T, and that's what's down here at the bottom. Now, what I do with forensics, the old-fashioned, not the stuff the genealogists use, the old-fashioned is just, I want to know how big a piece. I'm going to try to measure a particular piece of this chromosomal mess here. I measure it with chemicals that bind onto certain sites. And I want to know how long it is. How long a piece did you get from mom? How long a piece did you get from dad? Are they the same size or different? And I put those in a table. To do this, excuse me, I just copy the very few pieces, less than 1,000th of the genome or 10,000th of the genome. I'm just going to copy these pieces and they'll get visible. I'll make so many copies and I'll see how long they are. I have a way of measuring it by running it through a tube and seeing how long it takes. Okay, so I want those sizes. And these are the original 13 areas that the FBI told us to measure. And some of them have great names. So these are the chromosomes. Before people knew exactly what chromosomes did, they named them and they gave them the best naming possible. They called the biggest one number 1, the next one 2. They did make a mistake down here, but that's okay. So they're size order number. And some of these areas, these loci or locations, have really good names like D3, DNA marker on chromosome 3, S because it's only found there, it's a single location. And this is the 1,358th one measured moving away from the middle of the chromosome. Some of them have historic names like TPOX. It doesn't matter. I'm going to get 2 measurements from each of these. One, how long a TPOX allele just means form of the gene, how long an allele you got from mom, how long you got from dad. If I do that at these 13 areas, that's 26 measurements, I can identify you to the exclusion of everybody else in the population. So what do they really look like? Well, to explain this to grown-ups, I use Legos. And these are 4 colors. They correspond to the 4 types of bases, A, G, C, T. And I want you to tell me what's different. Here's, let's say, the chromosome from mom, this one here, and the chromosome from dad. What's the difference? Most of the chromosome is going to be identical because it codes for important things. But there's this area called short tandem repeats, or STRs. Tandem means one after another. And here's a short tandem repeat. This is a 2-base repeat. And so what would I call these? Well, I'm going to call this a 3 because there are 3 of that yellow, white, yellow, white, yellow, white. So I'll say mom has a 3 at that locus, 3 repeats, and dad has 1, 2, 3, 4, 5. Dad's a 5. So I would say at this location, on chromosome 1, let's say, the person is a 3-5. They got a 3 from one parent and a 5 from the other, but they're a 3-5. I do that at 13 areas, and I have you. So just a little table with 13 rows on it. So what's the procedure that we go through? Well, the first thing is we look for a stain. So are we going to look for touch DNA or a stain? You take a black light. You've probably all seen black lights, ultraviolet lights, and sometimes water will leave a ring because the dust migrates to the edges of the spreading stain, and you see a ring of dust reflecting. But semen in particular has a particular glow, but you can be fooled. And blood has a bit of a glow, and urine has a bit of a glow. And so if we see a stain, that means probably it's a biological fluid. Why don't we swab there? If you don't see a stain, you're dealing with low template, low amounts of DNA, what some people call touch DNA, that I don't like. Okay. And then you swab it with a Q-tip. You wet a Q-tip, rub the stain, and then you take a dry Q-tip, rub the stain, and you keep those two in an envelope together. And then you move the DNA from the Q-tip to water by sticking in a little test tube. And the test tubes are the size about my pinky nail now. And I cut the swab. I don't even use the whole Q-tip end. I cut a piece of the Q-tip and put it in there. And then I have to get the DNA out of the cells, and that's where that meat tenderizer and a little bit of detergent comes in, and the epithelial cells break open. I have to tell how much DNA I have because I want to use the right amount for the rest of my tests, and how much male DNA based on how much Y chromosome material is, because males have a Y chromosome, females don't. And then I'm going to copy those areas, like I said, those locations or loci, and determine the size of the alleles through something called capillary electrophoresis. Little tube. How long does it take each piece to get through it? The bigger pieces go slower, the smaller pieces go faster. I run some control sizes with them, and I can tell how big all the pieces are. Then I have to interpret the results. How many people are here? How degraded is the DNA? And then use some sort of statistic, because remember, consistent is meaningless. Calling it a match is meaningless. Now, all the male DNA in the world is a match to me. I didn't do all of those crimes. You have to have a statistic, and the statistics, you can give the same exact DNA data to two different labs, accredited good crime labs, and the statistic will vary by 15 zeros. Because there's a lot of assumptions that go into statistics, and the methods are a little different. But you have to give the jury something. I just want you to know that those numbers can be very different depending on who's doing the work. And then the report wording, as we talked about, the wording is very important. And then testimony, cross-examination, and other statements. And usually when I'm talking to a professional audience, I ask about errors and how they can be prevented. So here's an alternate light source, a UV light on some underwear from a case. And, the stains, I can't tell which of these stains are urine and which are semen. Like, as I said, sometimes semen has a certain glow, but a lot of times it's worn out and you just can't see it very well. But these are areas that you would swab or cut out and soak. Okay. So now problem 3. Now that we've cut our bit of the underwear out, we're going to try to do something called serology, and that comes from serum, blood, and it used to be blood typing, ABO blood typing. Now it's pretty much just tell me, is it blood? Is it human blood? And there's tests, there are screening tests that are cheap that say, oh, this might be blood. It also might be something else. But, that they're cheap, and so you cut those areas and try to get DNA. Or you can do nice specific tests in the lab with monoclonal antibodies, the same way you would do a pregnancy test or a COVID test. And they're pretty good for human blood. So it's not dog blood or fish blood. It's not maybe blood, maybe something else. But when you do a screening test for blood, how do you report that to the jury? So here's what happens in a courtroom. These tests are all, this is a Kesselmeyer test, also called the phenolphthalein test. It's a test for blood, but it's a screening test, and there can be false positives like horseradish, potato, turmeric, malt extract, cobalt, manganese, iron, lead, saliva, and pus are possible false positives. So you don't know for sure it's blood. It could be pus. And that's okay, because the way that I would use as a DNA analyst is I'm looking for where there is a place that might have a lot of DNA. So it's good to use a screening test. But if you're going to get into court and say, I did a blood test, that's blood. You know, there's blood on that hammer. Not there's my DNA on the hammer. There's my blood on the head of the hammer, like that, and I'm dead with a head wound. You know, that's important for the jury, the triers of fact, to know that something's blood, and that's what serology is. But now think about it. I said there's screening tests, and what are we going to call the screening test when we want to look for a stain that might be blood? We're going to call it a blood screening test, right? So what does the jury hear? Well, I looked at the items and I thought there might be blood. There was a stain. So I tested for blood. I used a blood screening test called the phenolphthalein blood screening test. We use it to indicate whether or not something can be blood. And it's a presumptive test. It's not a definitive test for blood. And so the jury's hearing blood, 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 blood. And then finally, the defense lawyer might get up and cross and say, well, couldn't it also be borscht? Couldn't it also have be a horseradish stain? This person, you know, they're German, they eat a lot of borscht or whatever. They eat a lot of stuff with horseradish. And couldn't it be that? And the expert will be honest and say, well, yeah, it could be. There are false positives for this blood screening test. So the jury has heard blood, 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 could be borscht, right? I asked the analyst, why not do it this way? Why not say, I did a screening test for cabbage, 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 cabbage. I did a fermented cabbage screening test. We looked for fermented cabbage. We found a stain that could be cabbage. We tested it, could be cabbage. Oh, in cross-examination, but it could also be blood. And the problem is, again, the language and the purpose of these tests is to get the right guy, to develop evidence for the state to make sure they got the right guy. And while that is a noble and a perfectly sensible task for for the prosecution of justice, it brought in a bunch of bias that people don't realize even that they're using. So by telling the jury it's a blood test over and over again, we have that problem. There's another problem that I'm grateful I think we've taken care of in Idaho. The presence of sperm, microscopy. You can see sperm cells under a microscope. They're the only ones with tails. I can teach anybody to do this. But sometimes the pictures aren't very clear. And they lose their tails. They lose their tails after a few hours, even in the vagina. So, you have to apply some stains to see them better. And when you have a lot of them, I mean, there's 300 million in an ejaculate. So if you have an ejaculate, you're going to have a lot of them generally. But you don't always have an ejaculate or a preserved ejaculate. So what the scientists came up with in the 70s was called a differential extraction. So now I have to teach a little bit about sperm cells. And what I teach my students is sperms have perms. So, you go to the hair salon and you smell that sulfur, that yucky smell. Well, it's the sulfur bonds that keep your hair in a curl. And so then they relax the curl. They break those disulfide bonds and reset whatever curl you want or straight, and then set the sulfide bonds to that, the sulfur. Well, the sperm head, very interested in keeping that DNA locked in there, has a really tough protein coat, and it's one of these sulfur-rich bondings. And so we use a chemical to break them open. What does this mean? It means I can get a vaginal sample from someone who says they've been sexually assaulted, and we can see if there's sperm cells in there. We have a way of breaking open all those epithelial cells I showed you, that with a little meat tenderizer and detergent, suck that liquid out, which has the DNA from whoever's epithelial cells there were, probably mostly the victim or complainant. And what's left? Those tough sperm cells. So I rinse them 3 times, and a lot of labs forget to rinse them 3 times. But you rinse them 3 times, and now you've washed away most of those broken epithelial cells from the man, the woman, whoever, and now you have sperm cells. Mostly, but there's always some carryover from that first group. And what do we call that second tube? So one tube has those epithelial cells mostly that have broken open. We put the DNA and suck it into a tube. Now we've washed these sperm cells. We break them open with that stinky sulfur chemical. And what do we call it? We call it the sperm fraction or the male fraction, but it's been called the sperm fraction in court cases all over the country. The problem is we're going to call it the sperm fraction, even if there are no sperm in it. Even if all you got was carryover female DNA from that first extract, we're still going to call it the sperm fraction. And why is that a problem? Well, if you go to court and there's just a woman in the sperm fraction, that was a problem for the prosecutors, right? Because the juries got confused, and I finally got some prosecutors to agree with me that, yeah, they should also join the fight to get rid of this term. But mostly I'm interested in the person who's on trial, who's against a system that has been built up to get the right guy and not to protect their rights as much. And so we've had cases where there's the man's DNA that somebody said is accusing of rape. The man's DNA shows up in that sperm fraction, but they didn't take a picture of any sperm cells, which they have cameras on all the microscopes. You can do this very easily. I have no idea why Idaho doesn't even, they don't take pictures. If they see it, they just write down a note of having seen it. They take a picture of my parking meter when I park wrong. You know, I can't understand why these lab analysts who have a computer with a camera on top of their microscope don't take a picture. But they'll still call that sperm fraction. And so what happens is, if you're getting leakage from that first epithelial, half of the time you're going to have a male if there's equal amounts, and half of the time you're going to have a female in the sperm fraction. If the male shows up in the sperm fraction, they're going to say it came from sperm. They're going to refer to it as the sperm DNA throughout the trial, only because they tried to do this separation, which works really well when you have a lot of sperm. But I've had a lot of cases where there's not so much. So finally, this shows you how dedicated some of these forensic lab people are. A group in Scotland and in Canada, working at two forensic science centers, decided to put this to the test. So how did they do it? These are their conclusions. I'll skip this for now. But this is, I'm going to show you how to read a result here. This is an electropharogram. Remember I said how there are locations. So here's a location in red up here. So this location looks like D6, I think it is. I can't tell. My eyes are not that great. But anyway, there's a location and there's two peaks. So that could be a person has two different sizes. What size are they? It looks like gosh, I can't read it. I'm so sorry. 17 and a 19 or something. I don't know. But there's two peaks. So this looks like it's a person. And the telling thing is right here, we look at a location that's called a melogenin. It's for a tooth enamel that is on the X and Y. And so we can tell if there's any male DNA, if there's more than one peak. So this one just has one peak. The electropharogram, that's what these prints are called, below has a little bit of male DNA. See the little Y there? Okay, so there's male. Now, what was the experiment these dedicated scientists did? They did not have sex with their partners for, I think it was 10 days. And then they had to have this kind of sex that the lab experiment said, which was the male would perform cunnilingus on the female. And that's the only sex that they could have. And afterwards, they would have the female put on a new pair of lab-provided underwear for various periods of time. So the idea is you had only oral sex, no sperm, right? It's been 10 days. There shouldn't be any sperm around. And we're going to take a swab from the underwear that this woman put on after having oral sex. And the question was, if we perform a sperm extraction, differential extraction on that DNA that the male only gave cheek cells, no sperm cells, are we ever going to see the male in that second sperm fraction? And indeed they do. That's what this is. The sperm fraction from the underwear of a woman who was given oral sex by her partner shows that sperm fraction has DNA from the male, and it's not sperm. And above the epithelial fraction, that first fraction, no male DNA. It used to be that I would go to court with a result like this, and I would say, yeah, that looks like they got sperm, because it's only in that second fraction that we see the male. Until I read this paper, and they said when you have low amounts of male DNA, some of the times randomly you're going to get male DNA in that second one. Don't be fooled by this. And so they set up parameters of how much to do. So I'm proud to say that Idaho does not use this sperm fraction name anymore. They still have some notation in their notes about it being male or something. But and I will say that my lab can take some of the credit for that, though labs all over the country, I think, are starting to switch. And I give this talk a lot. Some labs figured it out without me and just did it. And I love it when they're in the audience, because they say, has anyone changed this, and can tell the other labs why you changed it? Because I get a bit of pushback on some of this. Okay. And I won't go through more of those details. So in the old days, they used to talk about 1999, a differential extraction, trying to get sperm, and they would call it there were two DNA preparations, a non-sperm epithelial and sperm cell DNA. And they would take that sperm cell, and it would be extremely powerful to a jury if they found a man's sperm on a woman. His DNA might be explained. If I, change my child's diaper, my female child's diaper, my DNA is probably on their body, probably in their genital area. If my sperm shows up there, if a jury's told that's my sperm DNA and not just my DNA, we have a problem. So this is Idaho in 2018, sperm cell fraction you see in their report, and this is in 2022, fraction 2 is what they call it, the DNA obtained from fraction 2. So we're having some luck with this. Let's see, any other questions at this point? We're heading up to our time break.
Yes, we have about 10 minutes left. There was one question. Well, Darlene has her hand raised, so we'll let her ask it.
Okay. Darlene? Oh, sorry.
Yes. DNA testing, is it like how much you pay for it? Does that determine how good of a test it is, or is it the same across the board?
That's a really good question. So, and people will give you different answers. I'll say that standard DNA testing as practiced by the crime labs in the United States is great. And they have different kits they use, different chemistries. But for a simple case, like, everybody does it really well. The direct to consumer stuff, I just published a paper with a group of really brilliant people who, again, did most of the work. You get to be old, and you get to be an author on papers. Other people do a lot of work. Anyway, we looked at dog DNA testing companies. How well can these testing companies tell the history of your dog? And so we took pedigreed dogs who had records of several generations of matings that the AKC people believe, and then we randomized DNA to send to several companies. And what we found out was, and some companies asked for a photo. We sent photos to everybody, but we randomized the photos with the DNA. So you got a Saint Bernard's DNA, with a Chihuahua's photograph. And one company called the photo, they said it was Chihuahua DNA, even though we gave them Saint Bernard. We just swapped the photo. And that company, I don't think, is in business anymore. And it's not clear whether they looked at the photo or they just called it wrong. I don't know what happened. But what we did find was, so the AKC breed, if you have a purebred dog, the companies, the major companies, all got it right. What they had a harder time was the next generation back. Like to say that with a mixed dog, to say how much German Shepherd there was versus Pekingese or whatever, and they weren't good at that at all. So the question of how much money you pay, I'd say that's probably not the way to look at the problem. You want to look at, is the lab accredited? You want to go online and look at what people are saying about the results, but the accredited labs are usually pretty good. And then you want to price shop. So there's nothing wrong with price shopping. And they have specials like 23andMe was running these specials where it was like 1/10 the price. I hope that answers your question. Here's what sperm looks like when it works really well and you stain it with what's called a Christmas tree stain. But here's an actual case on the right. It was a jacket of a person, and that they only had one sperm cell. And there's no tail, and the staining looks okay, but it's an outside jacket, and so pollens can look like that too. And they ended up with a hung jury on that case. I'm going to skip that one. I'll just go to one other form of bias. I've published on DNA bias and in complex mixture interpretation, because there was a man who was in prison that we finally gotten out, where there was a mixture of DNA. It was several men who sexually assaulted a woman, and she recognized one of them from a high school yearbook, but couldn't recognize the others. The guy who did it and who admitted and whose DNA was all over the place, he gave them a name of names of two other guys. And that was introduced into evidence because the DNA experts said that those other two guys could not be excluded. And we showed the same evidence to labs outside of the state of Georgia, and almost all of them excluded the man who was in prison that I was working with. So we did a study on bias and published it. But I want to show you a more recent study by my co-author, Ideal Dor, on this. He looked at medical examiners. Now, as I said, we're an aging population in this group, I think. And I get notifications of my friends dying now. I lost 2 this last week. One of my friends, who I've already shown you, Christopher Tapp, died of a head injury in Vegas, and they released to the press that it was an accident. He slipped and fell, and I called the medical examiner and the homicide department, and my partner and the people who work for me were all concerned, like, they're not investigating this. It's a head injury in the back of the head, and now it's a murder investigation. So did we have an influence? I don't know, but I know that you can have a big influence. How do I know that? This is the study Ideal Dor did. He gave the same medical findings to 2 sets of forensic pathologists. These are medical doctors, special training in pathology, special training in forensic pathology, all who've testified in courts all over the country in terms of cause of death, manner of death. And with those same findings, he gave them 2 different stories. Half of them got this story with the white bars and half with the black bars. What's the difference? Of the ones who determined, by the way, whether it was an accident or homicide, and this means somebody's going to be charged, right? Of the ones who could determine it, they found differently under the 2 stories, the white story, the black story. Now, he published it with white and black bars. You'll see that there's 2 things he changed in the story. The white bars are the medical examiner with all the details was told it was a white child with their grandmother taking care of them when the child died. The black bars are black children with the mother's boyfriend caring for them. And you can see that the story of the mother's boyfriend, black child, the homicide is over 35%, and the accident is down at about 7%. And for the grandmother and the white child, it's just the opposite. Right? And so what's going on? Well, this article generated more letters to the Journal of Forensic Science than any in the entire history of the journal. So much so that the editor wrote his own editorial saying, we can't answer all of these. I've never seen anything like this. Usually these are polite debates. This was so acrimonious. My friend Ideal and his authors had to answer, I think, 20 of those letters, and, were threatened with lawsuits. And there were medical examiners who defended this, and I'll let you come to your own conclusions, who said they didn't touch the racial question, but one could apply the same reasoning to any question. They said, well, it's known that people who are unrelated are more likely to kill children. Now, first of all, I don't know that that's known, or how you would know that. That's a complicated study. But even if it were true, is it relevant? Well, we allow that in some places. We allow boys, for example, to be charged more for car insurance because as a class, boys are more likely to get in an accident. Though in my family, it was my daughter who got in the first accident. And should we allow it in this type of determination? Let's say it was established that people who are unrelated are more likely to kill. Should that be used in every, that means that that the totally innocent person who has nothing to do with the crime is suddenly more likely to kill that person than the people around that child who may be plagued by the child's difficulties, or the insurance money might be part of. I just don't know that that I buy that personally, and I have a strong disagreement with using that type of inference. But anyway, this is the case that I wrote about subjectivity and bias with the same co-author, Ideal Drawer. And we gave the evidence from our case, Kerry Robinson's case, to 17 analysts out of the state of Georgia at a accredited forensic lab. And out of the 17, 12 examiners said it excluded Kerry Robinson, the semen mixture from this multiple perpetrator assault. 4 examiners says it was inconclusive. Only 1 agreed with the original. That still wasn't enough to get Kerry out of prison. I wrote stuff for the New York Times. We had a number of exonerations using a new statistical package. Finally, the state of Georgia buys one of these statistical packages to analyze these complex mixtures. And they won't run it on Kerry Robinson's old case. First of all, they say they can't find the evidence. Finally, took us 12 years to get them to find the disk with the evidence or to make us a copy from the computer. And they wouldn't analyze. They bought this software called TrueAllele that could solve this and tell them Kerry's excluded. They wouldn't run it because they say, oh, we didn't validate it for the old chemistry. We use new kits now. And I was like, well, can't you try? I know you can try because it's just software. No, we can't do it. So I went to the author of the software, the guy who invented the software, the grandfather of probabilistic genotyping, Mark Perlin, who I got to know at a professional meeting. And I said, Mark, could you do this? Georgia won't do it, but they bought your software. And Mark did it, and he excluded Kerry, and so they had to let Kerry out. But that shows you that the arguments, as I tell my students, there is no proof, maybe in mathematics, there's no proof but conviction. You're convincing somebody that something is right. Always. If I have experiments, I'm convincing 2 out of 3 editorial reviewers that my experiments are sound and maybe it'll get published. And so conviction is how humans do things. It's what we believe is true. Now, in the most pure forms of mathematics, I think we can find exception to that. But let me just show you the last one that's Amanda Knox, who I'll be giving a talk with in Atlanta soon. I worked a little on her case, but she's become a very good friend. And I'll show you the last case because this is Charles Fain. Charles was in Idaho here, was freed in 2001 after 20 years. They say 18 years, but total of 20. For the wrongful conviction of a rape and murder of a 9-year-old girl in Nampa, up the road from here. And Charles got out before I moved to Idaho, but when I got here, I said, I met him and I said, we're going to try to get you compensation because all he was given was a pair of dungarees and a jacket for his 20 years in solitary confinement, being 4 hours from execution once. And a really nice guy. And then we also promised him and the police that we would help find the real perpetrator or somebody who matched the DNA. All that was left was a small piece of pubic hair found on the girl. And we helped the police and eliminated a bunch of people who they thought might be the suspect over the years. And then I was doing a video on the Hatfields and McCoys. Somebody was trying to sell something to the History Channel. And we were having fun out in Virginia, poking into graves and trying to get DNA from the soil. And the guy who had this tube to poke into graves, I asked him what he does. And he's like, I do ancient DNA in California. His name's Ed Green. It's like ancient DNA, like what? Oh, tusks and hair and stuff like that. I said, my God, do you think you could work with a fragment of a pubic hair that's 43 years old in an evidence room and get enough of these single nucleotide polymorphism SNPs to do genealogy, because there's no suspects. It's been this way for 43 years, no real suspect. And he said, yeah, I think I might be able to do it. So I introduced him to the Canyon County Sheriff's Department. They worked with him, and this is the first case Ed did. And I was like, have you ever done forensics? No. And now there's a company called Estrella. They're doing this. And this case is at trial right now. There's a guy who matches the DNA who, and I was hoping to be there today because a friend of mine who's retired from the lab was testifying. I wanted to hear her testify. But anyway, this will be the first case in the country that uses that genealogical type of DNA, SNPs, as criminal evidence. They also have other DNA evidence to corroborate, but they're introducing it for the first time. So all this Golden State Killer case and all these things you've heard about with genealogy, no one has ever introduced that genealogical DNA into a criminal court. They use it to find leads. And it's going on right now in Idaho, and it's part of our work. There's Chris Tapp and Charles Fain. We did get them compensation. That's Robin Long, who was my lawyer at the time at the Idaho Innocence Project. And this is where they announced the new method by Ed Green. That's UC Santa Cruz, a friend of mine and colleague. So, whoops, I'll just say that's my book with Calvin Johnson, who died this year. I'm flying to a memorial for him in a couple weeks. That's the story of his exoneration. He taught me so much about forgiveness and compassion and love and patience and faith. And I really enjoyed writing the book with him. I don't know if we have time for questions, but I'll sit. It looks like there's 30 things in the chat. Holy moly.
Most of those were just telling where folks were from, and they're from all over the country, from Memphis to Pittsburgh to New Orleans. So, and of course, some folks here in Idaho. But
all right, well, I'll just say, Sherry, endogamy is a really great concept, and yes, it affects our ability to use databases that don't accurately reflect a population that breeds with itself. So we have to make, if you're going to study Armenians who are, complain to be thoroughbred Armenian, you probably need to use an Armenian database, not just a Caucasian database. So that's a good question. Yeah, DNA gets transferred very, very easily. That's the whole principle. We swab it with a Q-tip, and now I can put that Q-tip on a test tube, or I can put it anywhere else I want. And that DNA, some of that DNA will be wiped off. So DNA is very easy to do. Yeah, somebody served on a high profile case, loving it today. I'm glad people followed it. Raleigh, I used to live in Raleigh. Colorado, Boise, good to see Boise, Albuquerque, Pennsylvania, Gainesville. This is great. Princeton, yay. Been to Princeton, love Princeton. Virginia, that's where I was doing the Hatfield McCoy stuff. Nice. Connecticut, hey, I was in Norwalk for many years, UConn, all my degrees. Fort Wayne, yeah, done casework there. Thanks very much. And I'm easy to find. Thank you for your taxes. Most of what I do is paid by the public. I am extremely grateful to have that trust in that position. And yeah, it's just a joy to go to work.
Great. Well, thank you so much. This has been a very enlightening and wonderfully complicated, as it should be. Frankly, that's part of the deal here. We see it on TV, and it all happens so nice and smoothly. And it's good to have somebody say, "Well, wait a minute." So thank you so much for that.
Use this. Anytime somebody's telling you about a gut, forget it.
Okay.
Know this. Yeah.
Thank you so much. I will let everybody know that the recording will be available on youtube.com under search for NKA Idaho, and under there will be all the classes. This will be one of them. And thank you so much, Dr. Hampikian. We sure do appreciate it.
All right. Thank you. Bye, y'all. Bye-bye.
Bye.